Um, wrong one. Okay, we're good, I think. Right, it's going and I actually think you don't have to come back if you, I can remember. It's the R and then I just put it on pause, right? I mean, if you want to, check back like 20 till or so, just to roughly, yeah, in case I messed it up for the second part of it. Okay, thank you, Jessica. All right. Hi, everybody. There's got to be a faster way to do this. Hey, there we are. I think I stopped at this slide. Right? Does that ring a bell? Can you all hear me if I leave this like that? All right. So did you have any questions from last time before we get going back to the liver? Okay. So I think I'd finished talking through this slide, right? We had talked about the 3 points about the liver, that it's the primary site for uptake of amino acids after a meal, primary site for essential amino acid catabolism, except for one very important group of essential amino acids. Which ones are not catabolized at the liver? Branch chain, very good, and then it regulates the availability of amino acids to the rest of the body, and we decided there's no storage depot, right? So this is my very abbreviated picture of enterohepatic recirculation. This has come up in MNT, and I'm giving you all in MNT this slide because it's just obviously not true to scale. But it's important to think about the fact that the portal vein is going from the small intestine to the liver. So if you're in the fed state, you've got a lot of amino acids coming in to the liver through the portal vein, right? What are the things that are recycled through enterohepatic? What are the nutrient? Well, there's one nutrient and then one facilitator of digestion, right? What gets recirculated that you're aware of through enterohepatic recirculation? Is there a nutrient that you're aware of? Say it louder. Okay, well, bile salts, right? That's not the nutrient I'm thinking of, but it definitely, bile salts get recirculated this way. Several times during a meal, in fact, right? We're very efficient at recycling our bile salts. So that's not quite the nutrient I had in mind. It's one that allows you to really have a pretty good long time before you start getting deficient in this. It's a vitamin. Probably you haven't, unless you've had this through advanced human, maybe you don't hear about this. You talk about storage of this vitamin for a very long time to vitamin B12. Yeah, great. I'm sorry, I should have seen your hand. So vitamin B12 is also circulated this way and retained. Very efficient. All right, so let's get into talking about the reactions with amino acids. Again, we're in the liver. We're going to talk mostly, we're going to think about the fed state at this moment because that's where we're at. We've just talked about a meal, amino acids coming in through the portal vein. So you remember transamination? Can I see some nods? Mostly some. So the way it works is you've got the amino, it kind of involves the transfer of an amino group from one amino acid to the alpha-keto acid of another. In this case, I'm showing alpha-ketoglutarate as a very prime suspect in many transamination reactions. There's an enzyme involved in the yellow with the purple. That's my enzyme signal to you, aminotransferase. This is just obviously a generic equation. When that amino group leaves this amino acid, it becomes the alpha-keto acid of that, shown by this R1 here. And then we make glutamate in this case. So this is a very common scenario, is that alpha-ketoglutarate accepts an amino group from some amino acid. It's why we called glutamate the intracellular hub of amino acid metabolism last time. Remember that? So glutamate, intracellular hub, because it's involved with a lot of transamination reactions. Note that this is always a reversible reaction. That's what I'm trying to depict here with the arrows going in both directions. And it also involves this thing called PLP. Anybody know the abbreviation, what that might stand for? You ever heard of pyridoxal phosphate? Maybe? Pyridoxal phosphate. Let me show you up here. And I guess I thought I had it written out somewhere. Where do I have it written out? Shoot, I don't have it written out. Shall I spell that for you? Do you know how to spell? Anybody know how to spell pyridoxal? You do? Pyridoxal, pyridoxal phosphate. Does anybody know what that's the active form of a particular vitamin? Do I need to do that? Spell that again for you? Pyridoxal, pyridoxal, and then phosphate, you know. Anybody know what the active, of which vitamin is PLP the active form? Pyridoxal phosphate. Anyone in the room ever hear of pyridoxal phosphate? I'm getting no's. Okay, so it's vitamin B6. So vitamin B6 has a really important role in amino acid metabolism because this reaction will not go without pyridoxal phosphate. And in my other life teaching metabolism, I would walk you through all the steps because it's kind of cool. Pyridoxal phosphate has the swing arm. It's like a crane, you know, and it takes the amino group from, anyway, I always picture it as if they're actually, you know, alive and cartoon characters. It's not exactly the way it works, but anyway, so what else? I am showing you now one of the two most common transamination reactions you're going to see. This one is the AST reaction. And so that involves aspartate. AST stands for aspartate transaminase. So right now you're going to start to, if this is not coming all flooding back from biochem, you're going to think about the 3 amino acids that are involved in the 2 most important amino acid transamination reactions. So the first I've told you about is aspartate transaminase. I've got the other one, alanine transaminase, on the next slide, right? So ALT and AST. So they involve 3 amino acids, these 2 transamination reactions, right? What are they? They're up on the screen, just, you know, thinking about that. What are the 3 amino acids that you're going to always see in AST and ALT, between the 2 of them? Aspartate, alanine, glutamate. Thank you so much. It's an obvious one, okay? But just I want you to think about that and the fact that the alpha keto acids of the other of their forms are also participants in these reactions. And they both have PLP. So remember again that they are reversible reactions, okay? That they can go in both directions. I'm showing them in the direction that they're going in the fed state. I'm writing them left to right to form glutamate, okay? Because that's what we're doing. In the fed state, we're making glutamate from other amino acids, including aspartate and alanine. Yeah? I'm not going to ask you structures, but you do need to know the reactions, right? So, okay, so in the fed state, we're moving in the direction of glutamate formation. And remember, it's a way of transferring amino nitrogen to glutamate. These transamination reactions are a way of getting amino nitrogen from aspartate and alanine, but we have other ones too. And I'm not showing you, there are a whole lot of other. So we have the big friendly giant upstairs. Or it's an explosion. Can we just be sure of that? It's not an explosion, right? Somebody's moving something? Okay, just to be sure. It's not something like, it sounds like fireworks a little bit. In that case, out the door we go. Okay, sorry, I lost my train of thought. So it's a way of transferring amino nitrogen from other amino acids to glutamate. Once we get into the glutamate, then that can feed nitrogen into the urea cycle. That's our next step. Okay, we're going to be going to the urea cycle in a moment. The other point is it's a way of interchanging and moving amino nitrogen around between one essential amino acid. Could be, you can use the amino group from an essential to make a non-essential amino acid, or we can make one particular essential, we can use one non-essential can make another non-essential amino acid, right? So it's a way of equilibrating amino acids to have them available, the ones that we need. Okay, sorry, there's one more point I want to make, so I'll hang on this one for a moment. And that is that different tissues have different complements of AST and ALT that's present in a lot of different tissues. Okay, both of these transamination enzymes, because we do amino acid metabolism in every tissue, right? So, liver, heart, and muscle are big tissues in terms of the concentration of transamination reactions, AST and ALT. We can use those clinically to help us differentially diagnose what might be going on with liver failure, for example. And I'm not going to give you too much more detail, or any detail much about that. But two little tidbits that might be useful to know is that ALT concentrations are much higher in the liver than heart. So after somebody has a heart attack, for example, AST, which is very high in heart, not so high. I mean, it's still present abundantly in the liver, but it's much higher in the heart. If you have a heart attack, AST tends to go up quite high, as opposed to, compared to ALT. So it just is a way of sort of helping us kind of know what tissue might be damaged, because when tissues are damaged, they release their internal enzymes, right? The cellular content of enzymes will spill. So AST might be higher after a heart attack. ALT tends to be higher if you've got liver damage, for example, than AST. Okay, my key points. So, anybody want to take a stab at why it's important? Why is transamination important? What have I said? Some of the points about it. Okay, a way of equilibrating amino acid nitrogen. I'll add to that. I mean, there are a few things, right? I said, I don't know, 3 or 4. So you can equilibrate. It's also very important way of dealing with amino nitrogen, getting it into the urea cycle after a meal, right? Primary way that we catabolize amino acids, right? When they come in after a meal through the portal vein. And that's probably the two most important points. Which transaminases are most active? I know this is a repeat session. It's called, mantra. AST, would you say, Kelly? And ALT, right? Those are the most active. And they involve 3 key amino acids, right? This is going to be on the test. It's just thinking about it. And then you'll be able to draw them out or recognize when they're drawn in the correct way, right? Which one is what we're calling the, intracellular hub? Okay, go ahead. I'm not going to, but I'm going to let one of your peers do it. Willie, you want to say? Glutamate. So this is a point of confusion. Glutamate, always remember that glutamate's the one that's involved in these kind of, this particular intracellular hub kind of transamination reactions. It's glutamate. We're going to talk a lot about glutamine, okay? So it will be confusing, so you need to kind of keep them straight. So transamination, and then we're going to talk about deamination in a moment. Glutamate is a way of getting that nitrogen from meal into the urea cycle. So that's the key intracellular hub, is glutamate. They are reversible reactions. Which direction predominates in the fed state to the formation of alpha-ketoglutarate or glutamate, if you had to make a choice? Glutamate, right? I've written it in the direction that you're going, because you're feeding glutamate is going to be getting that nitrogen into the urea cycle in a moment when we get there. Which vitamin is required? Vitamin B6, and what's its active form? PLP, pyridoxal phosphate. Okay, now a new one. Do all amino acids undergo transamination as their first step? This is a slightly different question than my question about, do all essential amino acids get degraded at the liver, right? That was sort of the first, that was the way we were talking about. No, not the branch chain. But this is a slightly different question. Do all amino acids undergo transamination? Do you know of any examples of ones that don't from your memory of biochem amino acid? I don't know what exactly you may have gotten in biochem. Are there any that we would say are highly conserved? So there is one, and it's lysine. And the reason lysine is conserved, highly conserved, it doesn't mean that it never gets degraded, but it means that it's really convoluted in terms of how it gets degraded. It doesn't undergo transamination for several steps, actually, in its degradation. So it tends to be one of those amino acids, essential amino acids, that we hold on to really well. So lysine is one that is an exception. There's another exception, another essential is threonine. Okay, so threonine and lysine they don't undergo transamination as their first step. They are degraded, at some point. It just isn't as readily, it's not as ready to happen or as likely to happen. One other point about transamination, and hopefully this makes sense to you. So transaminases have a high Km, which means low affinity. Right? Does that ring a bell? I love to see nods. So, right, low affinity for its substrate, which means, what does that mean? So after a meal, what does that mean for transaminase activity? What do you think? So after a meal, you're going to have high concentrations, right? And so what does that mean for transaminase activity? It's going to be high, right? So if you have a high concentration of amino acids coming through the portal vein, those transaminases are going to be active, right? Because they've got a high affinity, a low affinity but high Km. So at some point in the post-absorptive state, they'll fall off, right? Because the concentration will kind of drop. So that's another thing that helps us think about how the liver is sort of it takes a lot of those amino acids and degrades them, but only when there's an abundant supply, right? Otherwise, post-absorptive, then it starts allowing the amino acids to pass to other tissues, which is kind of critical, right? It's important that we have access to other ones. We just want to get rid of the surplus, if you want to think of it that way. Okay. The other major reaction for amino acids that you need to remember is deamination reactions. And this one is showing, I told you that threonine is one that doesn't undergo transamination as its first step. Its first step is deamination. It's a deamination reaction. So it can, again, requires PLP. It's an enzyme called threonine dehydratase. And basically the amino group comes off and we get alpha-ketobutyrate, which is its alpha-keto acid. And up here I'm just showing you that at the pH that we're talking about in most cells, the vast majority will be in the ammonium ion state. And that's because the pKa for that ammonia group, or that hydrogen, that extra plus, is like 9 or something like that, 9.5. So I say it as the same thing when I call it we say ammonia, aminonitrogen, just so you know there's some nuance there. Yeah, and we'll talk a little more about the meaning of that or why that's can be important a little later, I think. Okay, so the other major deamination reaction is the deamination of glutamate, okay? So remember that we had all those transamination reactions are making glutamate, then we've got to do something with the amino group of glutamate in order to get it into urea synthesis. So glutamate dehydrogenase produces a reducing equivalent in this reaction, yielding alpha-ketoglutarate and ammonia. Okay, and this is really important that this is happening because it's got very high coordinated activity. Glutamate dehydrogenase, whereas the transaminases are in cytosol and mitochondria, glutamate dehydrogenase is a mitochondrial enzyme. Okay, so just a tidbit there because we think about where this NADH goes, right? So if you're in the mitochondria, what might happen to that NADH? It can, what'd you say? All right, what's the purpose of that? Right, so we can make ATP from it. So that's helpful, right? If we're in the mitochondria when that happens. So the two-step process of degrading amino acids in a way to get the ammonia into the urea cycle has been termed transdeamination because it's a transamination reaction, forms glutamate. Glutamate then undergoes dehydrogenation to yield a reducing equivalent, and the ammonia goes into the urea cycle, and it's highly efficient, very, very useful. The first two steps of the urea cycle are also in the mitochondria. I think I've got some highlights here. Remember that we're in cellular, tissue speaking, we're in the periportal hepatocytes, right? The cells that are right around the portal veins. This is a very efficient extraction process. That's important as well. And we're in the mitochondria, and the first two steps of the urea cycle happen in the mitochondria as well. And that energy is helpful to have around so we can have that available for the urea cycle, the first two steps that require some energy. Any questions about so far? Okay. So now, whoa, that's a little weird. So taking the amino group off, we're going to talk about how we get rid of the ammonia, the urea cycle, and then we'll talk about what happens to the carbon skeletons after this. So this is the full slide of the urea cycle. We're going to break it out by steps. So you had this in biochem, probably, right? Did you have the urea cycle? Did you have to memorize it? You didn't. Okay, so each instruction, year to year, that differs. Okay. No one had to memorize the urea cycle before? Oh, okay. Well, we're going to memorize the urea cycle. But I'm going to break it down for you. And, it's because it's kind of interesting. I mean, there's this whole energy thing that's kind of useful to think about. So let me walk through it briefly here, because these are highlighted here. So step one, I like to just here on this slide just talk a little about sources of ammonia. So remembering again where we are, it's always important just that we're in the periportal hepatocytes. So we've got some ammonia. The first source we've already talked about, right? Ammonia is coming from amino acids, right, after they're transaminated and or deaminated. So that's a big source. We also get ammonia from foods we eat. There is some ammonia present in foods we eat. So that's one source. The other is that we'll get some recycling from from urea that comes back in through circulation. So urea, once it's produced, it gets into the blood, and then it comes back through. And so we get a certain fraction, it's a small fraction, that bacteria can work on in the ileum and, or not necessarily, yeah, in the terminal ileum, where you get ammonia released from that. It's urease activity, bacterial urease. And so why do I want you to memorize, think about the urea cycle, is because when we get to MNT, some of us, many of us in the room, we talk about what happens when the liver fails, okay? And I want you to think a little bit about it now because it does make it relevant to some degree. And so we get some production of ammonia from bacteria. And so that's a point that we treat. People with liver failure, we give them antibiotics to knock out those bacteria that are producing extra ammonia, because we're going to talk about it, but ammonia is toxic. That's why we have such elaborate systems to deal with it, to detoxify it as urea. And then we've got one more source that comes in to the urea cycle, and that's ammonia, that's amino group, or nitrogen, sorry, nitrogen coming from DNA, RNA, those purines and pyrimidines. We also have monoamines that we talked about a moment, last time we talked about monoamines like, the catecholamines and dopamine and other monoamine kinds of things. When those get degraded, we have ammonia coming from them as well. Oh, we have one more source. So we have kind of 5 major sources. The last, when we get to the intestine, we'll talk a lot about this, but we've already talked about how the intestine uses glutamine. So when it uses glutamine for energy, it leaves ammonia, right? And so that ammonia comes in through the portal vein, coming from intestinal metabolism of glutamine as well. I'm a little ahead of myself because I haven't shown you that that sequence of reactions. Okay, so we've got ammonia. Note that the carbon for urea synthesis comes from CO2. So where's CO2 coming from? Well, yes and no, right? Yes, it is coming from the lungs, but not necessarily into the intestine, right? You think about what the lungs are doing. We're breathing in oxygen, we're breathing out CO2. Most of that's going out. Do we get CO2 produced from some metabolism? Right? Every nutrient metabolism produces CO2. I mean, oxidative, the TCA cycle. And so we've got CO2 available coming from, and so when I'll ask it is, where does the carbon come from? And it comes from CO2, from metabolism. So we're going to join these two by an enzyme called carbamyl phosphate synthetase to make carbamyl phosphate, which is a 1-carbon entity. And the main step here, some things to note, is that it takes ATP. It takes this activator called N-acetyl-glutamate, or NAG, if you want to remember it that way. And it also requires magnesium. So do you remember from biochem, every time you have an ATP involved in a reaction, you typically need magnesium. You might need manganese, but you need that to stabilize the reaction for the ATP to be available for the reaction. And I will go back over these. So let's stay broader here. I got a little bit bogged there. Second step, you've got carbamyl phosphate and ornithine combining to make this entity called citrulline. And we were all in the first 2 steps of this. We're out in the mitochondria, or in the mitochondria. We're going to come out into the cytosol using the same carrier that carries citrulline out to into the cytosol. At the same time, it brings ornithine in. That's a dual carrier. And then we've got this combining of citrulline with aspartate to make arginosuccinate. That also takes ATP. How many ATP equivalents are at that step? Is it 1 or 2 or? So So I always make an assumption that you've had this in biochem too. So when you cleave an ATP to AMP, that's the equivalent of 2 ATP, okay? Because these are high energy phosphate bonds, right? If you go from an ATP to an ADP and release just one, that's 1 ATP. But if you go from ATP to AMP, that's releasing pyrophosphate. That's the equivalent of 2 ATP. Okay. And then we break arginosuccinate apart to yield arginine and fumarate, and then we get the last step where arginine gets broken down to yield urea and ornithine. So I'm going to go through the steps independently now. I'll click my animations here. For it to be a cycle, so we'll come back to this at the end, but this is the cycle part. The first 2 steps are the first 2 steps of the synthetic pathway, but it's not really the urea cycle in name, right? Until you get to where you are recycling the elements so that you can continue, right? So the concept of recycling, hopefully you've had that in biochem before and thinking about that. So it's an important, for energetic stability, it's a useful thing that it is, that we get recycling. And I'll walk through these steps independently, but the final step, so you can see ornithine is getting recycled up here in that final step, and then fumarate has to get recycled back to aspartate so that we can continue the cycle. Now let me go through the steps one by one. And I think I've said most of these points, so forgive me if I'm repeating myself too much, probably here. Let me get to my second slides. So if I didn't make the point well enough before, CPS1, carbamyl phosphate synthetase, absolutely requires N-acetyl glutamate, NAG, okay? Cannot function without NAG. It's an allosteric activator. And magnesium is also needed to help the ATP bind to the enzyme so that it can carry out the reaction. So we need magnesium, we need N-acetyl glutamate. The other point I made is the ammonia. The nitrogen is coming from ammonia, the carbon is coming from CO2 to make urea. And that's in the first step. The other major point that I probably didn't make before is that this step uses 2 ATP, right? You got 2 ATP going to 2 ADP. So two ATP are cleaved in that first step. Second step, and we're still back in the mitochondria, and now I've put in the carbon so you can kind of see what's happening here. I didn't put it here, but obviously this is 1 carbon. Carbamyl phosphate is 1 carbon. You combine carbamyl phosphate plus ornithine, you get citrulline, which is a 6-carbon entity. Ornithine transcarbamylase, you could abbreviate that OTC if you want, but it's helpful if every time you say that as you're learning it, just to say what the enzyme name is so that you can know what you're making. Ornithine transcarbamylase, you're taking ornithine and carbamyl phosphate to make citrulline. Step 3, we're in the cytosol now. This is where we get aspartate, which has 4 carbons combining with citrulline. We're cleaving the equivalent of 2 ATP here, and we're making arginosuccinate. So that's step 3. And so a point you want to write over here is this is where the second nitrogen comes from for urea. Urea has 1 carbon and 2 nitrogens. Okay, so the second nitrogen comes from aspartate. First one comes from ammonia, the second from aspartate. The other thing you want to know about this step is that this is the rate limiting step. So it kind of controls the speed at which the whole thing goes. And note again, because we've got ATP, we need magnesium to help that to stabilize that ATP binding to the enzyme so it can work. Step 4 is we're going to cleave that 10-carbon arginosuccinate to yield the amino acid arginine and fumarate, and that enzyme is arginosuccinase, right, which is an easy enzyme name because it's breaking apart arginosuccinate. And then the step 5, arginine gets worked on by the enzyme arginase to yield urea. So now we've got the nitrogen, carbon, nitrogen, you know, the structure that I wrote on the other slide. And it recycles ornithine. So now ornithine is available to go out on that, go back into the mitochondria on that carrier that was bringing citrulline out, right? And we can continue the cycle. So I've tried to make this as clear as possible to you all. Step 6, I think we're at. Yes, step 6, also very important, is that we've got to get this fumarate back to aspartate. So these enzymes should be familiar, I think. Hopefully you've had some through the TCA cycle and other enzyme reactions. Malate dehydrogenase, I'm not telling you fumarase, but that's the enzyme that cleaves that. So malate dehydrogenase, I think you've heard of before, right, with the shuttle system. And produces a reducing equivalent because it's a dehydrogenation reaction. You get to oxaloacetate, and guess what? We need AST. That transamination reaction helps convert the OAA back to aspartate. So remember, we talked about AST and ALT. Those are all, they're reversible reactions. And that reducing equivalent can be used to generate 3 ATP. Okay, so where are we net? So the net cost of making 1 urea is 1 ATP. So I'd like you to be able to talk through that. We cleave 2 at the first step. We cleave 2 at the rate limiting step. And recycle, or you know, we generate 3 ATP out of that reducing equivalent, and then it costs us 1. So it's a very efficient process, which is useful, obviously. Now, why did I have this? I think I just should hammer that. Okay, 2 minus 2 plus 3, you got the net cost of 1. Are there any questions about that? Okay. This is just walked out for you so that sometimes when I write things in circles and cycles, it gets confusing to people. So I've written it out linearly for those that have more linear brains and like to learn things stepwise, not to make it confusing. So choose the way that makes sense to you. Okay, there's the structure of urea. So, sorry, now I've got to get back to my my key points about the urea cycle. So, where are we going to say the C comes from? CO2. Where do the 2 nitrogen come from? Ammonia. You can say it out loud. And aspartate. Okay. How much energy does it cost? This is like rote learning. Sorry. 1, right? And you can explain how, even though you cleave 4, why it only costs 1. Where does ureagenesis occur? So first of all, where in the body? We got lost in the weeds, didn't we? Back to where are we? What tissue are we in? We're in the liver. What specific cells are we in? The periportal hepatocytes. Great. So periportal hepatocytes, those cells right around the portal vein. So thinking about what's going on that we're talking about, hopefully that makes sense to you. We're in the fed state. They're going to be coming in, gangbusters from the portal vein, and those periportal hepatocytes are going to take them up and metabolize them. And then where in the cell, in the periportal hepatocyte? Well, we're in the, for ureagenesis, right, we said the first 2 steps are in the mitochondria, and then the rest of the cycle is in the cytosol. Okay, so the first 2 steps in the mitochondria, the rest in the cytosol. Other key points are that fumarate and ornithine get recycled. And I hope you can appreciate that. Why is that important? Lindsay? okay? The fact that as we do that, we're generating the generation of an NADH that can help us get 3 ATPs. That's true. What would happen if we didn't recycle them? Wouldn't be much of a cycle, would it? Right? So it keeps the cycle going. The fact that we've got these important steps that recycle. How does it happen? I want you to go back, and so as you're studying, these are study questions, really. Okay. Now, what regulates the urea cycle? Well, we have a few things. So let me switch my notes here. So there are 3 primary places where, that we have regulation, really important regulation. So you can flip back. It's not right next door to this slide, but if you want to, you can think about this. What effect would you guess a high ammonia concentration will have on CPS1, carbamyl phosphate synthetase 1, which is the first enzyme, first step in the urea synthetic pathway? What do you think? It's going to increase, right? So we've got substrate activation there, right? The more the ammonia available, the more that's going to push that CPS1 activity. So that's definitely true. And then you already know this one. What effect does NAG, N-acetyl glutamate, have on CPS1 activity? So if we have a low concentration of N-acetyl glutamate available, what's going to happen to CPS1 activity? It's going to go down, right? I told you it's an allosteric activator. You got to have it bound to CPS1 to make it work. So N-acetyl glutamate concentration, it's important that that's high in order to have active CPS1. Now, there's an enzyme that makes N-acetyl glutamate, and it's appropriately or cleverly named acetyl glutamate synthetase. It doesn't have the N in there, but acetyl glutamate synthetase makes N-acetyl glutamate. That enzyme's activity is increased by a high arginine concentration. So where is arginine in the urea cycle? Where does that show up? Feel free. Switch back. I'm not going to do it because it'll mess my whole slide up, but I can't easily do it. Where's arginine? In the back. Someone in the urea cycle. Where does it show up? Yeah, please. The last step, right? So if arginine concentration is high, that activates the enzyme that makes the first step go faster. My simple way of talking about enzymes. Does that make sense to you? So it's kind of we call that feed-forward activation or something like that. The most important thing, though, is just that if that cycle is going gangbusters, because there's a lot of amino acids coming in, and you're making, you're getting arginine concentration. Arginine is being cleaved to make urea. As that goes faster, it's going to stimulate something that'll continue to make the first enzyme active. Okay, so we'll have more NAG available. Okay, so that's that first step. The other second step, ornithine transcarbamylase. This is simply substrate driven. So if you've got a lot of ornithine, high concentration ornithine, that's going to increase activity, right? And then the product, if you have a high citrulline, which is the end product, that will feedback inhibit OTC. I mean, this is pretty basic, right? You've had enzyme. Okay, and then the last point is that urea cycle enzymes and hepatic glutaminase, I need to be very careful how I say that. Urea cycle enzymes and hepatic glutaminase activities are coordinately regulated, meaning when the urea cycle is active, so is hepatic glutaminase. So at the end of all this, hopefully next time, we're going to talk about how coordination between tissues happens. So high protein intake, you're going to have high activity of the urea cycle enzymes and hepatic glutaminase, and that is part of the story about glutamine. So we talked about glutamate is the intracellular hub. Glutamine is the way that we get amino nitrogen around the body. And so in the fed state, you're going to get glutamine coming from other tissues that have been using and doing amino acid metabolism, including the muscle. Okay, we're going to talk about the fed state, and muscle produces a lot of glutamine. That glutamine comes to the liver, and it can be broken down and yield ammonia to feed it into the urea cycle. So glutamine is a way of getting amino nitrogen into the urea cycle as well, under conditions when we have both active, like in the high protein intake or high fed state. And we'll learn more about that in a minute. On the opposite side of that, in long-term starvation, you get a downregulation of the urea cycle and glutaminase, because we just, and there's more to that story about glutamine that we'll hit in a little bit. So any questions about the regulation of urea synthesis? Hopefully interesting enough that you can tolerate having to memorize the cycle. I'm sorry to make you. Lindsay. Okay. Okay. Yep. 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 Let me. So go back over that. Okay. So again, it starts with the fact that NAG is an allosteric activator of CPS1. So we got to have that N-acetyl glutamate around. So one of the ways that the body makes sure that that happens is that under conditions when you have active urea synthesis going on, you produce, you're going to have high concentration of arginine in those cells as we're going through the cycle. It's getting cleaved as well, but it is detected, okay, arginine concentration. We may also have other sources of arginine coming in, and I don't want to don't go there. I don't want to expand the story. Let me stay simple. So that high concentration of arginine, which is at the end of the urea cycle, activates back at the beginning, if you will. I mean, it's not really. It's activates the enzyme that makes NAG. Acetyl glutamate synthetase activity is upregulated by arginine concentration. High arginine concentration, high acetyl glutamate synthetase, which makes NAG. So then you've got more NAG around because it's bound every time you're doing this CPS1 reaction, it's bound to that molecule, that CPS1. So you need more NAG around to get more CPS1 to be doing the reaction. Hope I didn't confuse you worse on that. Is that clear? Okay. Any other questions? Okay. What am I doing with this? Oh. I guess I just have this again. I'm going to click this through. I don't know. You see that it's 6 steps. Okay. Seriously? Oh, I had a old slide. Forgive me. Okay. So this seems like kind of a no-brainer too. So I don't mean to be insulting to you, but it's not. Sometimes the most obvious things aren't always so obvious, right? So what happens, do you think, if a defect occurs in any of the urea cycle enzymes? So we get an inborn error of metabolism. They happen, right? So the urea synthesis enzymes, one or another, has a flaw. What's the most immediate effect physiologically that will happen when you have a flaw in the urea cycle? Yeah, Amanda. Perfect. Okay. We get a buildup of ammonia in the blood, right? Ammonia concentration will go up. So I've lost my track of what is, so let me click all the way through all of my questions, right? So these are kind of around the same idea. So if you have a buildup of ammonia, what does that do you suppose? And there are backup systems, so we'll just assume that, you know, we've got this high concentration of ammonia in the blood. What's the effect of that? Anybody, we haven't quite gotten there in MNT yet, but so say that you've got this high ammonia concentration. How would your body respond to that? What would you expect somebody to look like if they had that problem? High ammonia concentration. Not necessarily look, act. No ideas? Is it healthy for us? Okay, no. So what do you think the first or one of the primary manifestations of a buildup of ammonia in the blood are? Lindsay, do you have an idea? So let's not go there. I would not link that necessarily. Okay. The physiologic consequence, yeah. Okay, so not necessarily. I don't think that would help very much. Yeah. Okay, we probably would get a pH level change. What's the most, those of you, have you ever known anybody with liver failure? And anyone that's worked in the hospital before, or maybe you've had a family, relative or whatever. What kinds of changes happen in somebody with liver failure as you're talking to them, if you're able to talk to them? Neurologic, right? Neurologic toxicity is the first thing. Ammonia concentration is highly toxic, and it's highly toxic first and foremost to the brain, okay? So when we talk about liver failure, right, so I've just said, urea synthesis enzymes, well, we can take up some slack. I'm going to teach you about what we can do with ammonia at the liver if the liver is totally healthy, but if somebody's liver is not working well, this is kind of how we know how toxic ammonia can be, because it's really rare to have ammonia concentrations build up. We do not allow that to happen. The healthy body does not allow ammonia to be in any concentration at all because it's really toxic to the brain. It's toxic to lots of other tissues, in fact. So, my questions were probably maybe not worded quite as well as I could have. When we get to more about glutamine metabolism, I'm going to tell you about how we can detoxify ammonia in other tissues that don't have the capacity to make urea, right? So urea is vitally important because it helps us detoxify aminonitrogen, right? Because aminonitrogen is ammonia, and ammonia is toxic to the brain and neurologic, other nerve tissue and other things. And somebody with really significant liver failure, they're going to be in a hepatic coma if we allow that ammonia to build up, okay? So, any questions about that? Okay. Now back to how does aminonitrogen, and I know we're going to take a break, so I'm going to I'm close to finishing, I think. Yeah, we'll stop, maybe I'll stop after this last bit about amino acid nitrogen. So amino acid nitrogen, we've got this urea cycle. We said, the nitrogen for urea comes from ammonia, it comes from aspartate, right? That's kind of the localized idea. But of course, we've got other amino acid nitrogen that can feed in. So one of the ways is that we get transamination reactions that can help us to get glutamate. And I'm just, actually, let me just do my animations over here. So we have two places. One over here. Transamination reactions can feed other amino acid nitrogen into aspartate over here. Not really over here. It's, of course, this is paper biochemistry, but happening at the same time, right? So amino acid nitrogen can feed in through transamination reactions. That's really what I'm trying to depict over here. I'm showing alanine as one of them. And then we have glutamine and glutamate that are helping us get amino nitrogen, right? And so, again, anytime you've got transamination reactions to produce glutamate, glutamate can then be deaminated, and that's the immediate way we get from glutamate into the urea cycle ammonia. So let me just, one last, when we go through various tissues, I'm going to want you to start kind of putting it, I'll help you, I'll try to kind of remind you of how different tissues' metabolism of amino acids is helping us get that nitrogen into the urea cycle. One of those is that the intestine's use of glutamine, and again, I'm a little ahead, I don't know if we go there next, I can't remember, I think after the liver I do maybe go to the intestine, or it might be the muscle. When the intestine uses glutamine for energy, it produces ammonia, right? Ammonia comes in over here, but it also makes alanine. So alanine is also taken up from the periportal hepatocytes to feed into the urea cycle. So it's, again, the way tissues interact in the fed state helps to kind of make sure that we're detoxifying that amino nitrogen. That's my main point in saying that. Okay, I think I should give you a break. We'll get to the intercellular glutamine cycle is next, so I'll stop there. So let's take, you all like to have 10 minutes, right? Is that the normal? I will do the norm roughly, or maybe 9. Could we take could we be back at 8:45? Would that work? Okay, thank you. So see you in 8 or 9. All right, if we are ready, let me get us back to finish up what we have in this first section about the liver. Okay, so we've been pretty squarely in the periportal hepatocytes. So urea synthesis is one really major and important way that the body deals with amino nitrogen. Clearly, right? But the liver has, if you will, a backup system, and sometimes it becomes more important, more than just a backup, okay? But there's another way that the liver can deal with ammonia. And that is essentially what is happening in a different set of cells that are distant in location. And I'll show you a little diagram that kind of shows that in a moment. For now, I'm going to put them right next to each other, but know that they're distant. They're not right next to each other. And so what the liver has are these, in this other set of cells, paravenous hepatocytes. I think it's on the next slide. Well, it's a few slides from now. I'll show you in a second. But they're the liver blood supply, most of it, a lot of it's coming in through the portal vein, right? So the periportal hepatocytes are important. The other is central vein. And the central vein has these paravenous hepatocytes, the ones that are lining that central vein, okay? So any ammonia that escapes the urea cycle, right, it can be coming in from the gut through the portal circulation or whatever, that ammonia will be avidly taken up by the paravenous hepatocytes to protect the body from ammonia. So again, thinking about why this is important to the body, it's because we don't want ammonia circulating, right? So that ammonia combines with glutamate to go to the opposite reaction of glutaminase, which I guess I haven't really talked through yet. Well, let me say this. Okay. I mentioned glutaminase. Can we switch gears? I'm sorry to do that to you. Back up one step. Let's go back over here because I didn't really talk through that reaction. When I was talking about where does amino nitrogen come from in the previous slide, one of them was glutamine, right? And glutaminase activity, I said, is co-coordinately regulated. So when the urea cycle is active, so is hepatic glutaminase. And now you can see it the way I've written this here to help you maybe envision that. So when glutamine is coming in from other tissues, because we're in the fed state and we've got glutamine coming in to the paraportal, it'll get broken down by glutaminase to yield glutamate and ammonia. The ammonia goes into the urea cycle. Glutamate is also having its own deamination reaction, right? Glutamate dehydrogenase. So it's kind of a 1-2 step. Glutaminase and then glutamate dehydrogenase gets us to ammonia that gets into the urea cycle. Okay? So in the fed state, this is very active. But again, okay, so now I'll take you to the next, back to that. So what happens if ammonia escapes? And it does sometimes. It will, and there's ammonia coming in from the gut as well, you know, that gets taken up by those paraportal hepatocytes, you know, from the intestine. But any that escapes will go into this glutamine synthetic reaction. So now we're doing the opposite. Ammonia plus glutamate yields, this is very convoluted the way I've written it, I'm sorry, this is kind of the idea of, you know, glutamate and ammonia to make glutamine, and it takes energy to do that. It takes that cofactor magnesium to stabilize the enzyme, and the enzyme is glutamine synthetase. Okay, so this is an energy demanding reaction, but an important one because it makes glutamine, and then if it's going to go, glutamine then will go to the rest of the body. You know, it'll leave the liver through the central vein. But it's sort of the last gatekeeper for the liver as circulation is leaving through the central vein, that it will capture whatever ammonia might not have been made into urea. Okay? Now I'll talk a little bit about those enzymes. This is linearly written out. Glutaminase is breaking it down, and that's happening in the paraportal hepatocytes. Glutamine synthetase is making glutamine using energy, and that's happening in the paravenous hepatocytes. And so I asked the question, how does the location of these two enzymes, which are separated by tissue, incidentally they're also separated by intracellular location, but I can click back. I didn't tell you that. I didn't point it out. I wrote it that way. Notice that glutaminase is a mitochondrial enzyme. Glutamine synthetase is a cytosolic. This is obviously not built, not scaled, but just so you can kind of think about that. So there's a subcellular separation as well, even if they were to any large extent present in the same tissue, and often they may be in small amounts. Okay, but so they're separated by subcellular compartment as well as cell macro, right? They're distant. Sorry, now I got to click through. Oh, okay. So how does the location of these two enzymes avoid have you heard of this, a futile cycle, futile cycling? Does that ring a bell from biochem? Or maybe some other metabolism in this class that you've heard about? No, never? Okay. Well, futile. What do we think of when we think of futile? Useless, right? Okay. What would happen if these two enzymes were present in the same subcellular compartment and the same set of cells? What do you think would happen? Yeah, we'd be constantly reversing each other. We'd be making glutamine, breaking it down, making glutamine, breaking it down. Would that be a futile cycle? That would be a futile cycle. So the separation of these enzymes, both subcellularly, in case they happen to be, and probably they are to some small amount extent. So they're in different parts of the cell, and they're also in different cells with large activity in different cells. Okay, paravenous, such. So here's where I have a picture of that. Here's the central vein, and then the portal area is distant, and I think I've got a better, there's some distance between them, okay? And the central vein is as we're kind of leaving the liver, we can think about capturing that extra ammonia. This is just writing it out so you can see the reactions with the you know, the structures, all right? And so it's very repetitive. I'm just going to click through it just so you can see what they look like in terms of if you were to write them out. Here I'll stop and just say that there's an important other thing about glutamine synthetase, and that has to do with Km. So it has a low Km, which means what? Low Km means high affinity, right? High affinity for ammonia. So that's important because of where, you know, it's located. You need to have a good amount, I mean, essentially any ammonia that gets presented to those cells is going to be taken up and made into glutamine. And that's an important thing to protect the body from buildup of ammonia. Okay, so any questions about any of that? We're going to get back to that at the end. We'll talk a little bit about different tissues and how interorgan glutamine metabolism kind of works together. Okay, so we've dealt with the nitrogen. What about the carbon skeleton, right? So I said we were going to talk about both of those, which I had a long story about the amino nitrogen. So I've forgotten where my click starts. So yeah, I guess let's start on the fed state, fed side of things. So in the fed state, if we assume that carbohydrate energy intake is adequate to meet the brain's requirement for glucose, if we say yes, we're going to have high insulin, low glucagon. So you have learned about that, right? There's always this ratio, the balance between insulin and glucagon is going to dictate metabolic events, right? The next question is, are amino acids in excessive needs for protein synthesis? So if the answer to that is no, and you know we'll have small concentrations, then we're going to be doing protein synthesis. Again, we're in the liver. We're going to talk. I'll show you a diagram in a moment. If the answer to that question is yes, then we're going to catabolize, and a certain amount we're going to be catabolizing anyway too, right? But just the idea that a lot of that nitrogen is going to be going into the urea cycle after we've done what we needed to maintain proteins in the liver or whatever. So we make urea. The alpha keto acid in this condition, in the fed state, you're going to use some for energy, but much of it's going to go to fatty acid synthesis. We do have glycogen that we can make out of those carbon skeletons. So that's kind of the fed picture. And then I think I've animated over here in the fasted. We'll do this more simply. In the fasted state, low insulin, high glucagon, right? Because you don't need insulin to be active. This is going to tie in over here if carbohydrate energy intake is inadequate, even if you're in the fed state. So what would that be, a low-carb diet kind of a thing? People are at a very low carb, like 10 to 20 grams, you know, the Atkins at the beginning stages. So in that case, amino acids are going to come over here, and it's going to resemble what's happening in the fasted state. We're going to use the amino acid. We're going to separate out the ammonia. We're going to make some urea in the fasted state, but by far and away, the more of it's going to get made into glutamine. As I told you, in the fasted state, we get downregulation of the urea synthetic pathway, and we actually get more action at the paravenous hepatocytes making glutamine, right? And that kind of maybe, if you just think about it globally, it's not a lot coming in through the portal vein if you're in a fast. Now in this case also, the keto acid is then going to get used to make glucose. We can make ketone bodies out of it, and we'll generate some energy that way. Now over extended, you know, starvation, we adapt, right? So we don't break down or use a lot of amino acids over an extended period of starvation. But the initial one, we're probably going to be using, you know, a decent amount. I forgot what else is in this slide, so I'm going to click. Oh yeah, where are the amino acids coming from in this case? So if you're talking about in the fasted state, where are the amino acids coming from to be used for the purpose of gluconeogenesis and oxidizing some for energy? Where does it come from in a fast? muscles, right? Biggest source. It'll come from other body protein as well. We do break down some of our organ protein as well, but muscle is by far and away the largest tissue source. Okay, so back to this. Remember we were talking about this. So let me make sure that I'm very clear about this. All amino acids can be used to make fat. Okay? Most of them can be used to make glucose because most of them have some intermediate formed where it's a 3-carbon unit. And so most have some intermediate that is basically anaplerotic, adding carbon to the TCA cycle. One does not ever yield any rise in carbon. It yields only 2 carbon endpoints, and I'll show you when we get to the muscle how that works. That's leucine. Okay, leucine is the only one that is uniquely ketogenic. Which means it's ketogenic and not glucogenic. That's what I mean by when I say uniquely ketogenic. All the other carbons, skeletons can be used to make fat. It's not an issue. Is that clear? Okay. All right. So we've been in the liver, and I told you that this large fraction gets catabolized. We've talked about how that happens. I said not all of them get immediately transaminated in the liver, right? And so there's some small fraction, not that small, 20% or so, that leave unscathed. Large part, those are going to be branched chain amino acids, okay? But some others as well, just in terms of supplying low concentrations to the other tissues. So more than 70%, I'll say, are branched chain amino acids. So that's the answer to that. Which ones? Branched chain amino acids. We've also got some protein synthesis needs in the liver. Lots of proteins in the liver that we've learned about in MNT that are important to maintain under normal circumstances, the transports and enzymes and other proteins like that. We talked about this. Which amino acid is highly conserved because it doesn't undergo transamination as the first step in its catabolism, right? So it doesn't undergo transamination as the first step, and it was the one that had I said it took a lot of steps before you get to a transamination reaction. Lysine. So lysine is the one that's highly conserved, an essential amino acid. And then one last point about this before we leave the liver is that from fed to fasted, the shift between fed and then post-absorptive or whatever, plasma amino acid concentrations for the most part remain relatively stable. So there's a supply, an ongoing, a certain kind of sweet spot for delivery of amino acids around the body. The one that really goes up in the fed state is branched chain. Branched chain amino acid concentrations. So the leucine, valine, and isoleucine concentrations go up markedly in the fed state right after a meal. And that's because of this, the fact that so many of them are going to be leaving the liver to go elsewhere. Okay? But the rest, pretty much we maintain our concentrations pretty consistently to supply other tissues, and that's an important point. Okay, any questions about the liver? And I've tried to give you a few summary points, but I'm going to, in the interest of time, move forward here. Was that no, sorry. Okay, all right. So we do go next to the, oh no, this was Jillian. Oh wait, my daughter likes to make things interesting. Like, you're so boring. Got to do something with the animation. Okay. So I really should have placeholder. Yeah, I do. The intestine. Okay, so now we're in the intestine. This is kind of where we're going to help you later, just kind of encapsulate what we're going to do. So I'm going to try to focus on, so we're going to go back in time now, right? We've talked about the liver, and we're going to go forward, but we're going to go back first. And so let's just talk about the intestine really quick, because it helps, I think, to bring some of the points that I've already made home a little bit. So, the intestine, it's not pure philanthropy or whatever. Actually, what would the word be for, never mind. I'm not even going to go there. I'm obviously tired. It needs its own tissue to be maintained, right? It's letting a lot of it go through the portal vein, but it's going to keep some. And some of the stuff that needs to happen in the intestine, if you think about this, how often does the intestine, like an enterocyte, what is the life of an enterocyte? Roughly, how often do they turn over the cells of the GI tract? Sorry? It's like a day. Every day we get a whole new set, of cells, whatever. I mean, they're doing it at their own pace. So we turn over. We need to synthesize new tissue all the time. The intestine needs to be maintained. So it needs to be fed, right? When MNT, we talk about gut atrophy when you don't feed. If you go for a long fast, longer than a day or two, you can cause some gut atrophy to happen. So we need to be synthesizing proteins that help us make lipoproteins, and what's the one that's made in the intestine that's so important to absorbing fat? There's a lipoprotein that's made in the intestine. Fat. Chylomicrons, right? So ApoB48 is the apoprotein that you probably remember that from. Have you talked about fat already in this class? Okay. Anyway, and so that's one thing. The other thing is energy. So I'm repeating myself. I've said this before. Energy. So some amino acids, and particularly glutamine, are metabolized preferentially by the intestine. And so when I say the intestine, let me be clear that we're talking about the small intestine. Okay, I needed to clarify that from last time. We're not talking about the colon, the small intestine. So let me get to the next slide. So the small intestine absolutely needs glutamine. Luckily, it's highly abundant, right? It comes in very abundant amino acid and protein that we consume. So the primary energy source is diet. I'm sorry, I'm getting a little, I like to just say, yeah, actually, I'll leave it at this. So the other source is the blood. Glutamine is the primary nitrogen carrier in the blood. It is the way we get it around between tissues, okay? So blood glutamine is coming from a number of different tissues, but in large part, skeletal muscle, okay? So that's where it's coming from, under normal conditions. Big producer is skeletal muscle. We also make some in the lung. We make some in the liver, right? We talked about the paravenous hepatocytes just a moment ago. So we can make some, and under different physiologic conditions, the liver becomes more important as a glutamine producer for the intestine. It has a trophic effect on the GI mucosa. What do I mean by that? What do you think of when you think of trophic? T-R-O-P-H-I-C. Growth. Yep. So it helps to stimulate resynthesis and turn over cellular proliferation in the GI tract. And what enzymes does the intestine need to metabolize glutamine? You're going to see on the next slide, so you can come back and fill this in. Incidentally, I suppose I should, I don't know if this is confusing to say. Just so you know, the intestine, the small intestine, does use glucose as well. So about 20% of its energy needs comes from glucose, and then about 40% comes from diet, 40% comes from blood. Okay, so you can see a big fraction of the energy requirements are coming from glutamine. Okay, so this is busy, but hopefully there's some rhyme to the madness or whatever. I have a key down here. So I said glutamine metabolism requires two enzymes in the intestine. So hopefully you can see, first one would be intuitive maybe, right? Got to have glutaminase. And then you get glutamate formed. So in order to well, it's the thing that the intestine has is ALT, so it has a fair amount of ALT actually. And so glutaminase breaks glutamine down, releases ammonia. That ammonia we've already talked about goes through the portal vein up to the liver. And then glutamate can be transaminated by ALT to form alanine, and alanine also comes in large concentrations from the intestine. Ammonia is going to the urea cycle, right? So that kind of takes care of that. So our purple key compounds produced from glutamine metabolism, okay? So we've got ammonia, we've got alanine that get produced from glutamine metabolism, and then the starbursts, or the suns, or whatever you want to call them, are the compounds that are exported out. Those are the things that are released from the intestine, okay? So in this case, we've got ammonia. It's produced and it's released. It goes to the liver. Alanine, both. And then over here, we've got citrulline being produced. Where have we seen this before? Ornithine going to citrulline. In the liver, right? In the urea synthetic pathway. Oh gosh. So what does that mean? We have to have some enzymes from the urea cycle. Not all of the urea cycle, okay? So make sure you're aware that what we've got is some partial enzyme activity that's overlapping in order to use glutamate to get to ornithine. That requires other enzymes. And I'm not going to ask you to tell me how that happens. I'm not going to tell you. I'm not going to tell you. No, I could tell you, but I don't want to muddy the water. It's a focus. So ornithine getting to citrulline, what enzyme might you think is necessary to do that? Immediately. Ornithine to get to citrulline, what is it? Enzyme? Ornithine transcarbamylase, did you say? I'm sorry, I didn't quite. OTC is required. Now, the other thing that's needed, we need something else, right? There's something else missing. That's why you have to memorize the cycle, because it's kind of relevant to all the other tissues as we talk about partial cycles. And I know you haven't memorized this yet. So you need, first, you probably do. First, we need immediately, let's just talk about this reaction first. What's the other substrate in ornithine transcarbamylase? What is it? So go back in your slides, in your notes. I'm going to pull it out too, just so I don't. If you look at the urea cycle, OTC reaction, you can't just make citrulline out of ornithine. You need some other substrate, right? Can you see it? Somebody shout it out. Carbamyl phosphate. Thank you, carbamyl phosphate, right? So that's the immediate step. Ornithine plus carbamyl phosphate undergoes that OTC reaction to make citrulline. And I think, yeah, I have a picture to show you again, but those that we're thinking about, then you need the enzymes to make carbamyl phosphate, right? Because you don't just have carbamyl phosphate lying around. So I'm going to click forward, but I want you to go back. And as you learn the urea cycle, that will become obvious, right? Because you're going to think, okay, carbamyl phosphate, what enzymes do we need? So someone said magnesium, we needed ATP, right? The carbamyl phosphate synthetase, we also need the reaction. So let me just get to where I'm in my notes here. So from glutamine, we get ammonia. Some of that is going to go to make carbamyl phosphate because of that need to, because I showed you on this diagram, we're making citrulline is a major thing that we export. So we need all of the precursors and the other enzymes that are involved. And so the idea with glutamine metabolism is you've got the ammonia supply right there. So you can make carbamyl phosphate. for citrulline synthesis. Some of it will go also, through the portal vein to the liver to get made into urea as well. What happens to the alanine? It's also going to the liver for transamination reactions, oxidation. It can be used for gluconeogenesis under certain conditions. And then the ornithine. We've said where we've seen it before is in the liver in the urea cycle. What compound is made from ornithine, right? Citrulline. And what enzymes are required? You've got to think back to all of the steps that are needed to help you know how that works. Here's a bit of a trick question, not a trick, but if we need CPS1 to make carbamyl phosphate, right, which I've just said a moment ago, what other enzyme do you suppose we need in order to get the allosteric activator of that enzyme? CPS1. What's the thing that we absolutely have to get CPS1 activity? NAG, right? So you need acetylglutamate synthetase too. You need all of the structure, the foundation to make carbamyl phosphate, and then you can make citrulline. This is a really important question, or an interesting question. So we make citrulline, right? And where does it leaves the intestine? Where does it go, and what's it used for? Is in the next slide here. And here I'm finally showing you the enzymes reaction. So note that this is partial. Put a big star, circle this, okay? So the intestine does not make urea, right? It's just that it's got some of the enzymes. And the reason that it has some of the enzymes is that it's useful for the intestine to make citrulline. Citrulline will pass through the liver. The liver doesn't really do much to it. And it goes to the kidneys. And I'm going to just click this because it's animated in a weird way. So citrulline is used to make arginine in the kidneys. So I'm going to tell you ahead of time, when we get to the kidney, I'm going to show you that the kidney, actually I think I've got it on the next slide. I put it in the next slide just to show you. The kidneys have also a complement of urea enzymes, synthetic enzymes, but not the whole thing, right? So the kidneys can't make urea, the intestine can't make urea. They work together. It's inter-organ cooperativity. And the point of it is that this allows the kidneys to make arginine so that it supplies the rest of the body with arginine. Arginine is really important for growth, for protein synthesis. It's useful, right? We need arginine. And so this is a way for us to get around the urea cycle. We're basically sneaking away some arginine that we can have some fate other than immediate urea production. Because in the liver, that arginine is just going to get broken down by arginase immediately to yield urea. Okay? So the fact that the intestine does this, produces citrulline. Now I'm going to go to the kidneys. Okay, so we're deviating from the intestine. I think this is my last slide anyway. Yeah, it is. Okay, so just a sneak preview of the kidneys. The citrulline is coming in from the intestine, and it's got these enzymes, these two primarily. It has a small amount of arginase activity, but none to speak of in essence. And the reason, I'm sorry, okay, so the kidneys do not have OTC. There's nothing up here, right? So this blank is just showing you that it doesn't have those enzymes. It'll take the citrulline, it'll do these two steps, and it'll make arginine. And it might have a little bit of arginase activity. This is where paper biochemistry is cleaner than real life, right? So there is a little bit. But it gets immediately feedback inhibited by ornithine. And we're not making urea to any extent, okay? So when you get asked, do we make urea in the kidney? The answer is really no, okay? Because anything that gets made, this ornithine that gets made, very, very little of it is going to be there, and whatever small amount is there, it's going to feedback inhibit arginase. And so that arginine is then allowed to leave the kidneys into circulation. And that arginine then goes to other tissues for protein synthesis. Okay, so the primary two enzymes of the urea synthetic pathway that are present in the kidney are going to be these two enzymes, taking the citrulline and making them, basically converting to arginine. Is that clear? It promises to be confusing. It's a little, but it's kind of beautiful too. I mean, there's this, you know, intertissue coordination to allow for the production of arginine for whole body protein synthesis. Okay. My summary points, which I think hopefully will help you as you review. So any questions? That was a quick snapshot of the intestine, just the high points of what's happening. There are other things happening we could spend more time on, but whoops, I'm in the wrong thing. We have until, what is it? 35. Okay, great, thanks. Yeah, it's kind of important because you have a quiz next week, right? Is that right? Or Thursday? Thursday. What's today? Tuesday? Yeah, okay. Yikes. Okay. So I want to talk through the muscle, if I can. And I think I can. So we are back to our little diagram. We're going to come, you know, we've talked about the intestine, talked about the liver, talked about those branch chain amino acids, big numbers of them leaving the liver. And that's the answer to that question. I always get ahead of myself before I animate. So which amino acids are bypassing the liver? They're coming to the muscle, branch chain amino acids in large part, okay? And when they get there, some of them are going to be used for net protein synthesis and some are going to be catabolized, okay? And these are the structures of the branch chains: leucine, isoleucine, and valine. So hence, you know, the way we draw them is this branched structure. And I forgot where I said this, so let me just think about this. Oh, well, I'll say it here. Well, actually, let me just make sure. Yeah. Okay. All right. So now when we're in the muscle, there is a transamination reaction that happens in large part in the muscle. That is the first step. Branch chain amino acid transferase, or branch chain amino transferase. You'll see it written in various ways, but that's what this is, BCAAT. This is a reversible reaction, so I've tried to show it like that. It can go in both ways, and guess what? It involves glutamate, right? So the intracellular hub shows up for these, just like any other transamination reaction. And so that's the first step, and that happens in the muscle. The second step that can happen is the branch chain keto acid dehydrogenase reaction. And I was just going to say that this BCAAT activity is primarily in the muscle. It's both in the cytosol and the mitochondria, so it's kind of pervasive. It's everywhere, like any good transamination enzyme. The BCKAD is mostly under normal physiologic conditions, you know, where we're eating every 5 hours or so. Its activity is pretty low in the muscle. It is primarily a liver. We're going to talk about the second step. This BCKAD is a liver enzyme. Okay. So when we're, you know, again, normal physiologic conditions where we're eating regularly or whatever, the muscle has the BCAAT activity is going to be, you know, active as those branch chains come in the fed state, because it's like any other transamination. It's how much gets presented to it is how active that transaminase will be. The BCKAD activity under normal situation is not active in muscle, but it is in the liver. Okay. And then down here, and I didn't show you the structures of these. I don't think I have structures anywhere in this section here. But the reason that leucine is uniquely ketogenic is that it just ends up with 2 carbon 2 or 4 carbon units that get cleaved and used for energy, and it makes it very, so it can't be used to make glucose. Now the other thing about BCKAD that you should just stick away because it helps a little bit, I think for me anyway, it helps to think about subcellular compartments. It's a mitochondrial enzyme. In fact, it's very similar to pyruvate dehydrogenase, which you have heard about, right? You learned about pyruvate dehydrogenase. So it acts very similar, has the same cofactors, in fact, which is kind of interesting. Okay, so let me walk through it. Branch chain amino acid transferase, the cellular location, both cytosol and the mitochondria. We know it's a reversible transamination reaction, right? Reversible transamination is the type of reaction. And then BCAAT activity in muscle compared to the liver under normal conditions, much, much higher in the muscle than liver. And for our purposes, essentially, I want you to think as no activity. I mean, of course, it has a small amount for whatever it might need to do, but mostly we can say no activity in the liver under normal conditions. And that's important because it allows for a fate other than immediate degradation to branch chain amino acids in the liver, right? It allows those to pass through to get into circulation. So this is where that nuance of what I had said at the very beginning about the first step being transamination. Branch chains, it is the first step of their metabolism is a transamination reaction. It just doesn't happen in the liver. It happens in the muscle, okay? And then what happens to the alpha keto acids of the branch chains? I think I show you a diagram in a moment, but we'll just say here, just so it's complete, in the fed state, we can transport those branch chain keto acids to the liver where they get met by the BCKAD enzyme. So they can be, actually I'm getting ahead. I'll show you in a diagram in a minute what else we can do with in the fed state. In the fasted state, just to make it simple, a simple picture here, the fasted state, you're going to oxidize those in the muscle for energy. Okay, so in the fasted state, we'll further oxidize those for energy. Now, how's that happen? So branch chain keto acid dehydrogenase, I said, is a mitochondrial enzyme. The type of reaction is an irreversible decarboxylation, which is what a PDH, that is also that kind of reaction, oxidative decarboxylation, and it's irreversible. So oxidative decarboxylation is an irreversible reaction. And the enzyme complex that I said it's similar as pyruvate dehydrogenase. So what do you remember about that in terms of, you probably, maybe you didn't, I always ask this, I don't know that you learned all the cofactors. Did you see them ever? Did you get to see the reaction at all? So there are a bunch of cofactors. Do you remember kind of roughly, there's some vitamins, that's why I like to say it, because it ties in how other nutrients are involved. Anybody? Biotin? Which one? NADH, which is, what vitamin is that? Niacin, right? And I've got a picture in a second that will help you with this. So the other one that's involved in dehydrogenation reactions, the FAD one, is riboflavin. Anyway, I'll get to it in a slide. The other thing that you may remember about pyruvate dehydrogenase is it's regulated by, it's kind of highly regulated, right? Do you remember by phosphorylation? Or did you learn that? You might not have learned that, but anyway, it's very similar. So I'll show you the reaction in about 3 slides. So we'll answer that in the slide because it's easier to have you look at it that way. So what happens in BCKAD activity in liver compared to the muscle, BCKAD activity is much higher in the liver than the muscle under normal physiologic conditions. So it's kind of, they've got opposites, right? BCAAT is much higher in muscle than liver, the transaminase, and the BCKAD much higher in liver than muscle under normal physiologic conditions. And in a fast, what happens in starvation and also in metabolic stress, okay, which is those of you that aren't an MNT, what happens when you have an injury and you're reacting to that, or you have a major infection, your body is what mounts a stress response. You get a really up regulation of BCKAD activity in muscle under those conditions. So in starvation and in metabolic stress, you've got much more activity of BCKAD, the dehydrogenase, in the muscle. Okay? Something like a 5-fold increase. And that's important, and we'll get back to why, a little bit more on that in a second. We have a disorder, a genetic inborn error of metabolism that is a defect in the BCKAD enzyme. Has anyone ever heard of this by any chance? And it's called maple syrup urine disease. So it's an early infancy inborn error of metabolism in the BCKAD enzyme. And basically what ends up happening is you get alpha keto acids building up in the urine, and it smells like maple urine, which is kind of odd and weird, but that's why they call it maple syrup urine disease. Doesn't change the texture or anything like that, but it is certainly associated with mental retardation and death if it's not treated, and so we give specialized formulas that have low branch chain content to infants like that, and can help them manage to prevent problems. Okay, let me see if we can finish, kind of talk a little bit more about what's happening. So this is the muscle, and we're talking now in the fed state, and I know your slide is busy because I've got mine animated. So I guess we'll start over here. So we've got branch chain amino acids that come in from the blood, from the liver. They undergo BCAAT, yielding, these are the branch chains, they undergo BCAAT. They yield glutamate and the keto acids. And I'm using that same diagram, or that same thing where the sunburst is what gets moved out. And from glutamate, we also have ALT that will help make alanine. So similar in a lot of ways to the intestine in this way, that will, we have ALT yielding alanine as a major export product. The other thing that's kind of interesting is that the muscle, I told you the muscle is a major producer of glutamine, right? So it has to have the enzyme machinery to make glutamine, and that means it's got glutamine synthetase. Okay? So glutamine and alanine are the major export products from the muscle in the fed state. These branch chain amino acid, or these alpha keto acids of the branch chains, okay? And I could have written this as BCKA or something like that, but anyway, branch chain, the alpha keto acids of the branch chains, they can then go to other tissues where they could be reanimated, not animated, aminated. If they go to the liver, they're going to go through BCKAD because we don't have transamination there. Okay, so they can go to other tissues to be used, or they can go to the liver, and they'll be further oxidized there. So that's the fed state. Are there questions about the fed state? In the fasted state, wow, that was a big all at once kind of a thing. So we have breakdown, right? Protein breakdown will yield all three. Okay, there's another one over, leucine's over here too, but first we're going to talk about valine and isoleucine because they have slightly different metabolic fates in this case. So in a fasted state, starvation state, particularly earlier fast, branch chain amino acids valine and isoleucine will undergo the BCAAT activity, yielding glutamate. Same picture here, right? This is all the same, making glutamine, making alanine. The keto acids are over here. Those keto acids can now, because under these conditions, BCKAD activity increases 5-fold, as I told you, you oxidize those and you get these TCA intermediates. Okay, so we can oxidize those in the TCA cycle. We can use them in other ways. Under these conditions, leucine that gets produced from protein breakdown, I've written it over here because it gets BCAAT, and then it goes to the BCKAD, and then it immediately, because it yields acetyl-CoA and acetoacetate, gets oxidized very rapidly. And it produces a lot of energy basically supplies energy to the muscle, completely oxidized. And it yields ammonia that can then be made into glutamine for export. So the main thing is that under these conditions, BCKAD activity is increased 5-fold, and it allows for more complete oxidation of those. Under normal conditions, its activity is low, and it allows those keto acids to go elsewhere. Okay. I guess that's it. The next slide has your BCKAD reaction, and you can see thiamine as TDP, niacin, riboflavin, I can't even say it, pantothenate, lipoic acid, and that is basically regulated very similarly to pyruvate dehydrogenase, and we're not going to talk about that in any great detail. Are there any questions about the muscle? And I end at 35, right? So I think I will stop. That probably put you to sleep. I hope not. Maybe you can look over. We've done a little bit of storytelling today about different tissues, so maybe bring your questions next time. Yeah, Lindsay. Are you expected to? That's a great question. I have to remind myself what Xiaoli wrote the quiz. Dr. Chen wrote the quiz. So let me think about that for just a second. Did I print that? I have to think about that. I don't believe you have to memorize the urea cycle. I think it's something like you want to think about. I'm going to have her post in case I'm wrong about that. I did not ask, the things that you're going to be, like I'm not going to have you write out the whole cycle, okay? I want you to know the energetics about it. I've got a study guide that I've already posted with this material. Is it available? I think you need to memorize it in order to kind of be able to answer the questions as you go through the study guide, because otherwise you're just trying to pull it from the hat. And the idea, I'm trying to give you things that make sense to why you would want to know that. I mean, energetics are probably useful. Where does the nitrogen come from? Where does the carbon come from? Pay attention to my slides that have, those questions of what enzymes are involved, what amino acids are involved with AST, ALT. I can't remember exactly what she put in that. It's a little bit of a write-in quiz, right? It's a short answer kind of a thing, whereas the exam will be multiple choice, right? So if I'm wrong about that, I'll have her post something else, but just pay attention to all the slides where I give you questions. I think that's where she pulled them from. Okay, thanks for asking that. All right, thanks for your attention, everybody. Have a good day.